It's an honor to be here. It's exciting. This has only been presented once outside of Roche in a talk, and that was an hour ago. Second audience, and Matt and I will do this together. I'll give the background, and Matt will actually present the exciting data. So, as you know, Matt is the Director of Patient Partnership.
Something like that.
Something.
No, in fact, that is true.
So we have a conflict of interest. We both work for Roche. Roche owns the drug that we're going to talk about, so it's obvious conflict of interest. The drug that we're talking about is not registered, is not approved by the FDA or European agency. Just so you know, this is not an available treatment. It's an experimental drug. And very quickly, the background, because I know you've heard it several times in the last day. Lewy bodies were discovered in 1912 by Frederick Lewy, as Simon Stott told you yesterday, working with Dr. Alzheimer. Then not 10 years later, sorry, many years later, in 1997, it was discovered that the Lewy bodies are primarily made up of the protein alpha-synuclein, which you've also heard a lot about. This is the protein that forms clumps and can move between cells. And we're interested in the idea that the movement of synuclein from one cell to another could be one of the reasons that the disease progresses and there are more and more symptoms. So the drug we're talking about, which has the long, difficult name Prasinezumab, took me a month to learn how to spell it when we go on Roche. I actually misspelled it several times. I call it Prasi for short. This drug, we believe, its mode of action, it's an antibody, is that it mops up alpha-synuclein when alpha-synuclein is moving between nerve cells, causing the spreading of this Lewy body. So here's the drug, the green thing, binding the synuclein and getting it removed. That's the idea. And the trial we're going to talk about now is the extension of a trial that was published in August 2022, so just a year ago. This is the paper. Gennaro Pagano needs to be mentioned because he's the person driving this very much, a very talented neurologist, originally from Italy. The trial is complicated. I'll walk you through it, but we don't need to get into all the details now. We should focus on what's called the open-label extension, the long treatment, which Matt will show you. So initially, patients who entered this trial, there were just over 300, were either a high dose of Prasi or a lower dose of Prasi, or they were given placebo, which meant that every 4 weeks they had to go to the clinic and get an infusion of an inactive compound. And at the end of 1 year of treatment, the question was, did this slow progression? And the choice of an endpoint, and you heard lots about endpoints these days, how we measure the disease, the choice was a mix of UPDRS part 1, 2, and 3. And you may recall that parts 1 and 2 involve patient or caregiver reporting of how the symptoms are. Part 3 is the investigators rating motor symptoms. The trial did not meet its primary endpoint. I have to say that again. It did not meet its primary endpoint. So, inverted commas, it was not successful. It doesn't mean we didn't get information from it. And as you'll see, we're continuing to get information from it that are giving us hints of what's going on. But the primary endpoint, you can see that the blue line, people getting placebo, they deteriorated exactly as much as the two yellow lines, the two different doses of prasinezumab. The reason Roche didn't just at this point in time, which is now about 3 years ago, give up and say it's uninteresting, is that there were other things that were being studied. One was, as a secondary endpoint, the decline of motor function as assessed by the investigators in the 59 sites, including across the street at Spectrum Health. There was one site. The investigators assessed UPDRS part 3. There was a separation between placebo and the two treated groups, Prasi groups, after 1 year. And the digital endpoint, which is this app, which some of you might have seen, there's a table over by the other lecture hall, and we have representatives here who can describe an app where patients themselves perform things frequently in their home also showed a separation between the two, here in blue, prasi-treated groups and the placebo. So Roche thought maybe this is worth following longer, but it's not uncontroversial. I have to share with you, there are investigators, this is Alan Hone from England, who think that our trial and that trial of Biogen and Eisai actually marks the end of the road for monoclonal antibodies in Parkinson's, because both of them failed. We were invited to write a commentary in response to his editorial, Alan's editorial, who's a friend, a scientist, we can argue about things. So we wrote one which is entitled, "This is not the end for immunotherapy in Parkinson's," and we try to explain why. First of all, the drug Prasi targets certain types of aggregates of these Lewy bodies. It's possible that there are other types of aggregates that are important in the disease. Maybe we got the wrong one. That's a possibility. So making other types of antibodies that target other aggregates might be a good idea. That was one option. Or maybe it actually didn't work. I mean, we realized maybe it doesn't work. Another option is that it does target the right type of aggregate, but the drug which is given in the blood doesn't cross the blood-brain barrier sufficiently to get rid of sufficient numbers of Lewy bodies. We have no possibility of monitoring how many Lewy bodies there are in the brain of a living individual. We need to look at the brain in a microscope, and obviously we can't do that. So maybe that's one explanation. Another explanation, and again, think about the talks yesterday, perhaps it's not a good idea to measure parts 1, 2, and 3 together and lump them up, because parts 1 and 2, and this was early Parkinson's, they barely change. The total score you can get in part 1 is 52. In 1 year, the change is about 1 point, similar with part 2. So maybe they're not a good idea to include, whereas part 3, which is the investigator evaluating the motor dysfunction, that actually changes by about 13 points in 5 years. So it has a significant decline in a time window that you're going to see we're interested in. Then another thing is that we know, and you know, not every Parkinson patient is the same. So we're taking all these people in here. We're only excluding some people who have genetic forms. Maybe we should have looked at those who were more likely to progress rapidly based on other science and asked whether this drug is better in those who have a nasty version of Parkinson's. We didn't do that, but we did subsequently, and we have put a paper online, it's not published yet, we did subsequently analyze those that we knew were likely to progress more. And you can read this paper on, it's called Research Square. If you type in Prasinezumab and Research Square, you'll find it. And there we conclude that if we take those more rapidly progressing individuals, there's actually quite a big separation in motor dysfunction. Prasi actually does look like it's slowing, but it's a subgroup and it's not so many individuals. Finally, this is where Matt comes in. The other possibility is that the treatment period was too slow. 1 year is not a long time. There's not a huge amount of deterioration in 1 year. So smart people at Roche, before I joined Roche, said, okay, we have the signal and the digital endpoint with the app. We see UPDRS part 3 is looking interesting. Doesn't mean the drug works. I'll say that again. It doesn't mean the drug works, but what if we followed patients longer? And that's what was done in an open label extension. So I'm just about to hand over to Matt. So the first year was the paper with a primary endpoint. There was a second year when everybody, also those who were getting the fake infusions, went on Prasi. And in this part, we were looking at, is there a difference between those who started getting Prasi on day 1 versus those who started getting it after a year? And actually, again, in UPDRS part 3, there was a difference. But at the end of this was all planned. There was the idea that the study was going to be over. Patients would go off drug for 3 months. They would come back in and be evaluated for safety. And they'd be evaluated if there had been an effect here, had it washed out, had it disappeared. So the smart people at Roche decided, let's follow these individuals. Those who want to, you see 271 wanted to out of 310, let's follow them up to 5 years. Come in every 4 weeks. It's a lot of work, you can imagine. Follow them up to 5 years and ask the question if a longer treatment actually results in something that might be benefit. We didn't have controls, right? We don't have placebo anymore, so we had to take what's called real-world data, and we took this famous group of patients that Fox Foundation has funded, the PPMI, Parkinson Progressive Motor Markers Initiative. God, thank you. And the PPMI data set, we pulled out 303 individuals. Some statisticians at Roche did some massaging and converted the 303 people into 324.16 people. Don't ask me how that's done. See, the people that really matched these people were the ones that got more weight in the analysis. They spent weeks doing this. So we asked, will they progress differently? But what happened was COVID. So COVID came along. So when this study was going to start with this long 5-year treatment, it was in 2021. Contracts had to be signed. Research nurses maybe weren't there and doctors weren't there. So instead of 3 months washout, on average, we had about 6-7 months of washout. That means patients did not go to the hospital and get their monthly infusion. This is a very important point. And we have now 18 months of this time window followed up. So some people here have had the drug for another year, some have had it for much shorter periods. But we're now following them and we're focusing on, handing over to Matt, UPDRS part 3 in off. So this is the condition, no drug overnight, doctor or research nurse looks at the patient. And this is what we find.
Thank you.
That's the one. Thank you. There we go. Thank you very much, Patrik. So yeah, very interesting. And as we say, we've got 18 months worth of information already, still continuing on. You also have to bear in mind that it always takes us a little bit of time to analyze things and see what's going on. So 18 months was actually like a year and a bit ago. So right here today, it would be, I think, something like, where are we at now? Almost 48 months on open label extension. So there's the next round of data that's getting analyzed, doing every year analysis now. So Patrik showed you a couple of graphs beforehand, which showed the placebo arm, which of course we don't have anymore, because we're now having the external control arm, as we say. So real world data arm, people who are participating in PPMI, which we've matched. We are magic statisticians who convert 300 people into 324. The way that they do that, just to say, is they've really identified people from the, 36,000-ish people who are in PPMI. They found the people who've got a similar age, they're the same gender of the people, they were taking the same medicine as them, they were diagnosed around a similar time. So they're finding people who are almost like for like, except they've got a different name, live in a different place, sometimes see a different doctor, sometimes see the same doctor. And this is our light gray line that's moving on. So this is people as they progress over the course of 12 months, 1 year, 24 months, 2 years, 36 months, 3 years, right? Over the course of the study. And so what we see is at the beginning, everyone's sort of the same. And then the lines go in slightly different directions. Do you have any idea what could explain the lines going in different directions?
Sure.
Say again?
They were off the medication.
They were off the medication, yeah.
So
Social and moving around after COVID?
Could be, yeah. Maybe, yeah. So, I mean, COVID happened around this point right here, washout period for us. And you do see things started to go up a little bit more. People weren't possibly getting all the exercise that they wanted to. We've got a couple of different ideas of explanations. So one of these is, so we should probably say, this is a positive thing that this line is not going up. This means that people aren't moving along the UPDRS Part 3 scale. It means people's motor progression is not getting worse. One of the things that could be happening is you get to go every 4 weeks, you get to go for an infusion. You're seeing a doctor, you believe you're getting a medicine. It could be something that we call the placebo effect. right? So the belief that you're going to get better helps you get better. Well-known principle within Parkinson's disease, which is why we always include the placebo arm, right? That could be. You could also have a bit of investigator bias in theory, right? So that's the people doing, the doctors who are seeing people with Parkinson's every time. You think that probably that tends not to be the case. There's more than 50 doctors involved in this study. And for every single one of them to have introduced their own bias, probably not likely, but we can't definitely say that that's not possible. We are going to have a bit more data at month 48. That's our next cutoff. It's off the end of this graph right now. And that's also going to be interesting because we've got some imaging happening at that point. Other things to mention, Patrik really likes the phrasing that's come up on this slide. So I'm going to read it verbatim. I will read it verbatim because otherwise I'll be told off, right? So it has not escaped our notice that the limited worsening in prasinezumab treated participants suggest a possible slowing of disease progression due to therapy. Or simply said, maybe the drug works, right?
Do you know where that comes from?
Oh, Patrik encourages everyone to Google the first part of that phrase sometime. Well, you can say what it is. Oh, Patrik.
So Watson and Crick discovered the structure of DNA in their classic Nature paper in 1953. This was the final sentence. They wrote, "It hasn't escaped our notice that the pairing of bases in the DNA could be suggests a possible mechanism whereby inheritance occurs or something like this." And it's considered the greatest understatement in biology. Because they knew they had discovered how DNA replicates and so on.
It might be.
Necessarily say the same thing for prasinezumab yet, right? Because one of the other things that we need to remember is these results are exploratory. We need another clinical study to confirm or deny this. Fortunately, because of some of what Patrik was saying earlier, the fact that we had a digital biomarker that showed us something, the fact that we had seen on part 3, even if we had officially a failed trial, the combination of part 1, 2, and 3, but because we saw something in part 3 a couple of years ago, we launched a clinical trial called PADOVA, which is currently ongoing. And there you see we've got 500 people with Parkinson's have joined the study. People are receiving either prasinezumab at the low dose. Like, why the low dose? Well, over the course of this trial, we saw that there wasn't much difference between high dose and low dose. And with low dose, you overarchingly in medicine's development, you see fewer side effects. So if we can get the same benefit with fewer side effects, that's a positive thing. Low dose is in. And the primary endpoint this time, it's the part 3, where we're seeing something occurring in the first trial. So now we've got to wait time to go on to see whether this turns out not to be a figure of our imagination, and actually something that can be confirmed. So very excited to see that. And as I said, we also have more data coming out of this trial ongoing because people are still being really great and donating their time and I mean their family's time to come every 4 weeks and come and get medicine from their investigator and continuing to contribute their data. And we've actually taken out the data cutoff at 48 months over the summer. So we're currently crunching through that, future presentations at future conferences will show that. And I think we're also very excited because something we heard about yesterday, remember DAC-SPECT, the dopamine transport imaging? We've got that imaging at 48 months. So one of the questions yesterday that someone asked was, can you see a difference between someone who has a DAC-SPECT in, shall we say, 2000 and then 5 years later in 2005, do you see a difference? Yes, you usually do. The question is what difference we'll see. We're all excited to find out, but even we haven't seen it yet. So we're sitting there waiting, chomping at the bit a little bit. With that, huge thanks to everyone who's taken part in Pasadena. Time, family members, investigators, all of our colleagues at Roche as well. They've all been working very hard and continue to do so. With that, we're finished for this time. Hopefully we'll be back with some more information in the future. But we can answer any questions if you've got any.
The baseline on the left, the 2, 4, 6, that's based on, is that a percentage or what?
This one.
Yes,
So this is the score change on MDS UPDRS.
so it went up 2 points, 4 points.
Exactly,
Okay.
from wherever someone's baseline was.
Okay.
So maybe someone started at a 3 and another person started at a 1. It's talking about that change.
So basically in a 4-year period, you're showing the group that's tracking through the Fox Foundation of gaining 8 points in 4 years, whereas potentially
Potentially the half.
2.5 points.
Exactly.
So it's like a half gain, which is big over time.
The interesting thing is, for the first year, they follow each other pretty much exactly.
So do you think that's because you need a buildup?
Well, there are two interpretations. One is that it is, or that we're just seeing placebo effects. And of course here,
Okay.
there is a placebo control group
Inbound.
and they follow that. I mean, but the more likely in my mind is that there is a need for a buildup to get rid of these Lewy bodies, if that's what the drug is intended to do. Doesn't happen overnight. It's a gradual process.
But the actual problem that causes the Lewy bodies still continues. You're just cleaning up the mess.
Yes, exactly. And if you remember the lovely analogy that someone made yesterday about our pod of strawberries with mold on it,
Yeah.
right? You're stopping the mold spreading to another strawberry rather than getting rid of the original moldy strawberry.
The original moldy strawberry might have occurred 20 years before.
Okay.
So the original moldy strawberry might have occurred over here.
Yeah.
And we can't turn back time. But now with the new biomarker that you heard about yesterday also, it might be possible, and this is looking way into the future, to screen people at, let's say, age 50 by doing either, well, currently it's a lumbar puncture, but in the future, maybe a blood sample. And you're going to say, hey, you have a bit of moldy strawberries in your blood. If we have a treatment for these, we could then apply the treatment before, and I'm talking now 20 years down, right? We could then apply the treatment and prevent disease, but that's a long time down the road.
It's interesting that Michael J. Fox was in Back to the Future and traveled back in time. So maybe we're gonna go round circle. You buy the DeLorean, John? Hey, you able to measure the elimination? We'll do what we have.
So that's a great question. So can we measure elimination of alpha-synuclein? When this study started in 2019, the seeding assay wasn't really established. It was being done in laboratories. So there aren't samples on all the people who went into the study. There are some people where there are samples and there's analysis going on this. However, my guess is that we're not going to see complete elimination because the way that assay currently works is you just need essentially one aggregate in your sample, one single one, and then that could amplify the outcome of this assay. And you talked about it, Martin, I think. Did you talk about the seeding assay?
I mean, the smallest amount, but we also had other presentations.
Sorry, Simon spoke about the seeding assay. So Simon spoke about it and ERAS. So the seeding assay is essentially an all or nothing. It's binary. Either you have a positive or you don't have. In the future, I think there will be a quantitative one, but that hasn't been developed. And then one could see in the future whether it's possible to reduce the load of aggregate.
Maybe we could go back in 5 years and look back on that small group of people, a subgroup of people where they've regularly donated the CSF. We could go back and see, are we seeing an increase or decrease? Right now, all we can say is they have synuclein.
The Alzheimer's field is fortunate because they have PET scans, brain scans, where they can see the amyloid in Alzheimer's, visualize it. And then when they've developed their new emerging drugs, is that they've been able to see this amyloid disappear in a living individual. We can't do that.
Do you have any explanation for why the UPDR1 and 2 were not significant and the UPDR3 was? And what does that reflect as far as what people are reporting?
So Patrick's prepared a slide anticipating this question. Actually, if we just click through the, how do we do this? Oh, there we go. I don't do Macs.
So this is a different study, a different group, but it's the same patients. It's the PPMI patients. Samantha Holden, who published this 2018. So here she's plotted the scores over 5 years. And you can see part 1, it moves from 5 to 10, or 5 to 9. Part 2 moves.
1
per year, right?
Yeah.
Between 5 and 10 over 5 years is about 1 point per year.
So I'll ask you the question, how is that possible? Because the maximum score here is in, I think, 52. So we're only going about 20% of maximum in early first 5 years. The maximum for part 3, which is a much more sensitive scale, is 130. So they go up to about 33, which off the top of my head is about a quarter of, that's a 25%, so a little bit more. What is the difference between these? And these are really the investigator, the nurse, or the doctor writing down things. These are reported by the individual. And we talked a little bit, I was in and out of this room yesterday, about how difficult it is to know if you're worse now compared to 6 months ago.
Don't you think we adapt too, and so we don't show that as progression? Then you
Right. So also the living situation might change because as time goes by, during this 5-year period, it's not unlikely that people due either to age or due to problems with the disease will retire. So when you answer a question like, I forget the exact wording, but in UPRS part 1, there's something about do you feel socially impaired, or can't you go out and meet people, things like this. Of course, if you're in a working life, you may actually say yes, because I have problems going to work. But if you're retired, you may actually say no. So you may actually improve on the scale when you retire. And apathy is another measure here. So, you know, the inability to engage and so forth. And it's been shown that the inability to engage is actually coupled to problems with cognition and being able to perform tasks. So what we call executive dysfunction. So they've seen that people who have executive dysfunction, they don't even notice that they have apathy themselves. So we have a bioinformatician, Ben Ribber, who said to me the other day, Patrik, I think if we get a drug that improves executive dysfunction, it's going to lead to worsening of apathy because patients will know they have apathy. That just gives you a whole sense of how complicated this is.
So is there room to have a loved one's reported measure included in trials going forward? Because rallying 2 years ago, we did a wonderful survey where certainly Team Spark, who drove that survey, had identified that they were really accurately reporting on their loved ones living with Parkinson's symptoms, with the exception of a tiny bit of intensity on a couple of symptoms. But the accuracy on what is happening with that individual was really phenomenal. So is there room in future clinical trials? Yes, exactly. I'm pointing.
CDP, what do you say about this? So what the FDA views as a relevant outcome is a very hot topic. And
yeah, so I think one of the challenges that drug developers and that everybody is challenged, is dealing with is this, part 3 versus part 2 versus part 1. The FDA came out and with, patient-focused drug development. Right? They say if you develop a drug, it also I can.
Okay. When you develop a drug, it also needs to be relevant to the patient, right? Because and they have the same. Yeah. And they just have a famous example, right? Which is part of the part 3 examination is finger tapping, right? So you do finger tapping, and you could develop a drug that, and for Parkinson's is a little bit smaller and not as fast, and you could have a drug that improves that, and maybe you go a little bit back to normal. But if you don't care about it, that you now all of a sudden tap your fingers a whole lot faster, or what is more important to you is your anxiety or your apathy or your, other, symptoms that you're experiencing, other than motor symptoms. What have we gained, right? Then you have improved finger tapping, great. Finger tapping.
Flamenco dance.
Or yeah. So
So of course that is a point as well, right? But Patrik, lays out some of the problems with that as well. Measuring what matters to the patient is really, really difficult, and many of these different aspects that, for example, Patrik brought up as well, they're really hard to incorporate in a measure, right? So I think collectively, as a field, this is what we're, this is the challenge, right? How can we develop something that is relevant to the patient, right? That it fits in this patient-focused drug development, and that we can measure, right? And I'll be honest, we're not fully there yet. I'll make the call, and I spoke with Matt about that yesterday as well. I'll make the call again to the field. This is data needs to drive this, working together, coming together as the field, the drug developers, the academics, and especially people living with that experience of Parkinson's disease need to inform, what is important, how can we measure, success of a drug. And that could be a combination of these kind of measures and measures that, really relate to non-motor symptoms, and to your, functional impacts, right? How is, your tremor impacting your daily life, for example? It's challenging. And we don't, I know, or the drug developers think, well, maybe the critical path has the answer. We don't have the answer. We need to collaborate on that, and together with all relevant parties, including the health regulators, because it's a challenge for everybody. Nobody has a plan.
Well, what Helen said is a great concept, that the partner or the spouse will be able to do the reporting. However, not everybody who's affected with Parkinson's has a partner.
Yeah, absolutely.
Number two, the relationship between the patient and the spouse partner will affect the scoring. So to imagine that the spousal scoring is objective, that's not true. And I'll give you an example. There are scientific papers about the sleep disturbances in Parkinson's, which obviously affect the spouse. Because if there's waking up in the middle of the night, unless I'm the spouse, because I wouldn't wake up if my wife woke up. Most people wake up when their spouse wakes up and get disturbed sleep. When the spouse or the partner gets disturbed sleep, do you think they will rate non-motor symptoms differently to when they've had a good night's sleep? So it's very complicated, not trivial.
Out of interest,
It's a big challenge for the field.
I always bring up the diversity card at the moment. Do you have any difference between gender, male, female? Because I read a wonderful paper that basically showed that access to medicine or access to healthcare system is very different if you are a man with a wife versus a wife with a husband. Because typically the wife pushes the husband to go and get diagnosed earlier, pushes them to go to every single appointment, pushes them to make sure you do that. And the husbands aren't quite as good at that overall. And so I'm wondering, but I would be I'd love to see that but yeah, we can solve it, but we need to work together.